Hello students, welcome back to the classroom point. In our today's class, we are going to discuss the electrochemical cells, in which we are going to discuss one of the electrochemical cell, Daniell cell, the first part of the electrochemistry. Now let's begin what is electrochemical cell. Electrochemical cells, also known as the galvanic cell or voltaic cell. Here the diagram is about the Daniell cell. What is electrochemical cell? Basically, electrochemical cell is the one which can convert the chemical energy into the electrical energy. So we can generate the electricity from the chemical reactions happening in the cell. So now let us see here galvanic cell construction or else Daniell cell. It contains two beakers. In the left hand side beaker, a zinc rod is dipped in the zinc sulfate solution, and in the right side of the beaker, copper rod is dipped in the copper sulfate solution. And these two rods are connected through the metallic wire. Here ammeter shall be there. This point we shall discuss later. Here zinc rod acts as the anode. There copper rod acts as the cathode. So now let us see what is going to happen in both of these electrodes. So whenever zinc rod is connected to the copper rod, a potential difference arises between the zinc rod and the copper rod. So what is the potential difference and how it is going to arise? Here zinc rod dissolves into the zinc sulfate solution at anode. Oxidation reaction takes place always at the anode. So zinc metal atoms, whatever present on the zinc rod, they try to dissolve into the zinc sulfate solution. That means zinc atom in the solid form which is on the metal rod loses 2 electrons and goes into the solution in the form of the Zn2 plus ions and 2 electrons. So these 2 electrons, that means zinc metal atom before dissolving into the zinc sulfate solution in the form of Zn2 plus ions, it leaves its 2 electrons on the metallic rod itself. Like that, many metal atoms, when after dissolving into the zinc sulfate solution, finally this metal rod is accumulated with the electrons. That's why it gains the negative charge. Hence this anode is represented by the negative sign, which is called the negative electrode. And these electrons starts flow using this metallic wire. This flow of electrons is shown by the arrow mark. So arrow mark is tilting towards the copper rod. That means electrons flow is happening from the zinc rod to the copper rod. Finally, electrons reaches the copper rod. So copper rod in the copper sulfate solution, Cu2 plus ions and SO4 2 minus ions are present. Copper sulfate is blue in color. This Cu2 plus ions, now the reaction is taking place at the cathode. At a cathode, always reduction reaction takes place. Cu2 plus ions, which are present in the solution, take up those 2 electrons coming from the anode, and finally copper deposits on the copper rod. So copper ions starts depositing on the copper rod. So in this process, after when this particular process happens for a long time, we can see that more Zn2 plus ions starts dissolving into the solution and making the solution almost positive in nature. So that here more positive ions accumulated over the surface of this electrode. Why? Because electrode is carrying the negative charge. So that these positive ions get attracted to the negative electrode and start depositing on the surface of the anode. And here copper rod, after Cu2 plus ions start depositing on the copper electrode, so that it is left, solution is left with the sulfate ions. Sulfate ions carry the negative charge, so that negative charge start accumulating over the surface of the copper rod. So when this accumulation of the charges happens, then the cell stop functioning. Reason is, we know that in this particular process, zinc is dissolving by leaving the electrons. That means zinc 2 plus ions has to dissolve into the solution. Zn 2 plus ions carry the positive charge. Here charge accumulated over the surface also positive charge. So positive charge repulsion occurs, so that a further dissolution of the Zn2 plus ions do not occur. That means generation of the electrons will be stopped. On the other side, sulfate ions, negative charged particles, are depositing on the surface of the cathode, and this cathode actually electrons it is giving to the Cu2 plus ions. That means the electrons has to enter into the solution through the cathode. So electrons are opposed by these negative charges, so that electrons also will not try to enter into the solution, so that process also will be stopped. So both of these processes stopped because the circuit is not finished. So to avoid this accumulation of the charges, inverted glass U-tube is connected to the both electrolytic solutions. So this is called the salt bridge that contains agar-agar solution, agar-agar gel, along with any of these compounds, inert electrolyte, KCl or KNO3 or ammonium chloride or ammonium nitrate. Any of these inert electrolyte can be used. What is the purpose of inert electrolyte? Inert electrolyte, whatever you are adding here, that should not react with chemically either with any of these electrolytic solutions. At the same time, it should not participate in the deposition of the, that means electrode reactions, it should not participate. And one more thing, both ions in the inert electrolyte should have the same speed, so that their conductance capacity almost should be very equal, so that whatever the conductance capacity is coming from the one side will be balanced from the other side. And both of the ends of this salt bridge is completely closed with the cotton. Now, by adding this salt bridge to these electrolytic solutions, K plus ions, for example, if I take the KCl, K plus ions travels into the solution and attract the negative charges, and Cl minus ions travels into the solution and attract the positive ions, so that these ions are finally will be kept away from the surface of the electrodes. So electrode surfaces becomes free, and the reaction now again begins, and the cell starts functioning and produce the electrons. The opposite of the flow of the electrons is known as the flow of the current. So current flows in this direction. Now here copper rod, why it is shown in the positive sign means, here copper rod continuously losing the electrons, that's why it is shown as the positive sign. So this is the representation of the, that means Daniell cell functioning. Now the question is, what happens if the Daniell cell is connected to the external source? Here three conditions are there. This is external battery. So if external battery is having voltage less than 1.1 volts, here we need to discuss one more point that I forgot. Daniell cell, this is able to produce the current of voltage 1.1 volts. So this much voltage it can produce. Now if this Daniell cell is connected to the external source, which is able to produce less than 1.1 voltage of the current, then the cell functions normally. That means a dissolution of the zinc occurs at the anode and deposition of the copper occurs at the cathode. Electrons flow takes place from the electron flow always from the anode to cathode, or negative electrode to the positive electrode. In this case, zinc electrode to the copper electrode. Current flows from the copper electrode to the zinc electrode. And the ions movement also in the salt bridge, positive ions goes that side, negative ions come this side. Now, if it is connected to the external source whose voltage is equals to 1.1 volts. If external source voltage is 1.1 volts, see the Daniell cell voltage and external source voltage both are same. That means both are in the static equilibrium now, so that there is no other area where this current can be sent. That is the reason why here no reactions happens at both the electrodes, and no flow of electrons also observed, and this ammeter or voltmeter, whatever we have taken, that shows exactly its points upward direction. That means no movement of electrons are observed because both are having the same potential, same voltage. Condition number 3. If external source is having voltage 1.1 volts, means now battery capacity is more than the Daniell cell, which is able to give the more current than this battery. Then more current is coming to the less current, so that the current enters into the cell. When current enters into the cell starts functioning exactly opposite what we discussed earlier. Earlier we discussed zinc rod acting as anode, copper rod acting as cathode. Now when that voltage is higher, then copper rod acting as a anode, zinc rod functioning as cathode. So copper rod now functions as anode, and here zinc rod functions as cathode. Now Cu2 plus ions don't deposit on the copper rod because now it is acting as anode. Anode means oxidation reaction takes place. So copper metal from this copper rod starts dissolving into the solution in the form of Cu2 plus ions, so that the copper leaves 2 electrons on the surface of the copper rod. So now it becomes the rich source of the electrons, hence it is shown as the negative electrode. Now electrons flow is from the copper rod to the zinc rod, so that the movement of electrons shown by the arrow mark like this. That means the voltmeter or the arrow mark tilts towards the zinc electrode, and here zinc deposition do not takes place. Zinc Zn2 plus ions deposition takes place, or dissolution do not takes place. Why? Because now it is acting as a cathode. So at a cathode, Zn2 plus ions in the solution take up 2 electrons coming from the copper rod and deposits as the zinc metal on this zinc rod. So since electrons are taken by the Zn2 plus ions, it is acting as a positive electrode. So exactly that means flow of electrons is in this direction, and the current flows in the opposite direction. So this is the functioning of the Daniell cell or the galvanic cell. Now can we construct some more cells of these Daniell cells? Means as we can take again anode, here cathode. Means now coming to the concept of again external source less than 1.1 volts or no batteries present. Let's come to the normal Daniell cell, so that by placing so many of the similar patterns of the that means cathodes and anodes in a sequence manner, we can construct the number of Daniell cells in a sequence. So in this case here, starting we discussed the potential difference. What is the potential difference means? Now when Zn2 plus ions dissolves in this solution, the charge differences arise here. Here negative charge is accumulated. So when copper is deposited over here, here also negative charges are deposited over the surface of the electrode, so that here also charge difference arises, so that is having the deficiency of the electrons. So when this is happening, whenever you connect, automatically electron starts flowing from the anode to the cathode in the normal Daniell cell. Over the period of the time in the normal Daniell cell, zinc rod dissolves and the copper rod size gradually increase. Why? Because Cu2 plus ion starts depositing over the surface of the copper rod. So this is about the Daniell cell normal functioning. So in the next class we shall discuss about the how to represent this cell and cell reactions along with the potential. Thank you, one and all.